So the next thing is what happens to the pyruvate. Once we make pyruvate, what happens to it? So there are four different pathways in which pyruvate can go. Two pathways are reversible, so we can make pyruvate to one substrate and pyruvate to another substrate. But two pathways are irreversible, and this irreversible pathways happens in mitochondria. Similarly, the two reversible pathways are happen in cytoplasm. So the location and then so the first pathway is the reversible cytoplasmic pathway in which the pyruvate is converted by an enzyme known as alanine transferase enzyme with the help of vitamin B6, which is also known as pyridoxine or doxin. For the purpose of remembering the pyridoxine name, that the X is replaced by 6, so it has this common syllable or common sound which it makes, that pyridoxine is the enzyme which helps alanine transferase to make pyruvate to alanine and alanine to pyruvate. And this happens, so alanine transferase make alanine, which obviously inhibit the pyruvate kinase enzyme. This alanine helps the muscle to liver transportation of different amino groups. So this alanine is carry amino groups and to transfer those amino groups from muscle to liver. And the next uh, reversible pathway of pyruvate is in the cytoplasm, which is lactate dehydrogenation pathway. So pyruvate is not only converted into alanine, it can also convert into LDH or lactate dehydrogenase enzyme. From this name we can assume that the end product is obviously lactate, and the it is dehydrogenation, so there is an involvement of NADH or electron. So NADH means we are using vitamin B3 niacin, which can make NADH2, and we, for the sake of remembering, we just write it as N3 or vitamin B3. So vitamin B3 helps lactate dehydrogenase enzyme to convert pyruvate into lactate, especially whenever the body is suffering from anaerobic conditions. Whenever there is intense exercise, whenever there is increased level of workout, then we need energy, so we convert pyruvate to make lactate in order to attain that anaerobic respiration. And this anaerobic respiration is pretty much common in RBCs. Anaerobic respiration, it is in RBCs, in WBCs, in kidneys and testes. And this anaerobic condition is very much whenever the person is suffering from the increased level of LDH, the increase LDH or increase in anaerobic respiration is shown in whenever the person is suffering from heart failure. Whenever there is heart failure, they cannot deliver hypoxia, so RBC hypoxia. There is no blood, so the tissue is suffering from ischemia, and there is whenever there is infection. So for infection, WBCs are involved. For heart failure, the kidneys and heart, both are involved. The hypoxia, RBC is involved, and ischemia, whatsoever tissue is the ischemic tissue. So whenever there is these conditions happen, then there is increased level of LDH. Whenever we see abnormal labs, whenever there is LDH, then it is either hypoxia, either heart failure, either ischemia, or either infection. So this is the second pathway. So the two pathways which are happen in mitochondria are carboxylation and dehydrogenation. So on this, the next pathway is pyruvate. First we discuss pyruvate carboxylation. So in the mitochondria, irreversibly, the pyruvate get carbon dioxide to make pyruvate carboxylate or pyruvate carboxylation. This pyruvate carboxylation, it gets carbon dioxide from this. The name carboxylase, it means the enzyme is adding carbon dioxide into the pyruvate. So after after the pyruvate is getting carbon dioxide, with the help of an enzyme, with the help of a vitamin known as vitamin B7, also known as biotin, it makes oxaloacetate, and this is happening in mitochondria, and it is irreversible. This pyruvate carboxylation enzyme, it actually making oxaloacetate. This oxaloacetate is helpful in the neogenesis of glucose or gluconeogenesis. So whenever we are doing gluconeogenesis, or whenever we are in the fasting state, we are using pyruvate carboxylation enzyme, carboxylase enzyme, to with the help of biotin, to make oxaloacetate. And this pyruvate carboxylase is not only in gluconeogenesis, but also it can give constant supply of oxaloacetate, also TCA, constant supply of oxaloacetate, in order to move that cycle. So the last pathway is very much important and very much tested, is what happens when the pyruvate is dehydrogenase. Whenever the pyruvate is dehydrogenate or dehydrogenate, then dehydrogenation mean we are making NADH, and this dehydrogenation is done by the help of vitamins 1, 2, 3, 5, and lipoic acid or lipoate, lipoic acid or lipoate, vitamin B1 thiamine, vitamin B2 riboflavin, B3 niacin, and B5 is pantothenic acid. So these are the vitamins which act as a cofactor for this whole complex. So pyruvate dehydrogenase is known as a pyruvate dehydrogenase complex, because it involves not only one enzyme, but it involves a lot of factors. So it is a very huge complex. So this pyruvate dehydrogenation, it can convert pyruvate into acetyl CoA. This acetyl CoA not only inhibit dehydrogenase enzyme, but it also inhibit carboxylase enzyme. And this acetyl CoA is very much important in the TCA cycle. Yes. So whenever there is a transition of pyruvate into the TCA cycle in the fat state, in the glycolytic state, then it can uses pyruvate dehydrogenase enzyme with these complexes, to make to move that TCA cycle. So it is important in fed and fasting state. So now we have a pretty much 2 or 3 conditions. What happens when we don't have pyruvate dehydrogenase enzyme? So no pyruvate dehydrogenase enzyme means we don't have TCA cycle. So whenever there is no pyruvate dehydrogenase enzyme, we cannot use TCA cycle, we cannot make acetyl CoA. So no acetyl CoA. We have known that acetyl CoA is not only used in TCA but also it is used in ketoacidosis. Also it is used in fatty acid synthesis. So acetyl CoA has a lot of function. So whenever the person is not having the TCA cycle, the person is not having the pyruvate dehydrogenase, it cannot make acetyl CoA. So that pyruvate is built up into the body. So there is increased level of pyruvate, which is actually neurotoxic. It can cause symptoms which are neurological deficits. So the pyruvate is shunted either into the lactate dehydrogenation, either into the carboxylation, either into the alanine transferase. So we can see increased level of lactic acidosis, increased level of alanine. The increased pyruvate, so increase ALT levels or alanine, increase LDH levels or lactate, which can cause lactic acidosis. So there is lactic acidosis, increase anion gap, and there is increased serum alanine. So how to treat that condition? We just treat by giving ketogenic amino acids, which are leucine and lysine, which can make ketones, and the body is never ever undergone the pyruvate. The body is not forming pyruvate because pyruvate is formed by the gluconeogenic enzymes, gluconeogenic amino acids. So we are restricting that pyruvate, we decrease that pyruvate. So if we decrease this pyruvate, we automatically decrease lactic acid, ALTs, and LDH. So we use ketogenic leucine and lysine, which automatically run the rest of the cycle, which automatically give the acetyl CoA, which run all of the rest of the cycles. So whenever there is a pyruvate dehydrogenase deficiency, we use leucine and lysine. The next thing is arsenic poisoning, and I will describe arsenic poisoning over here, because arsenic poison, it inhibit one of the cofactor of pyruvate dehydrogenase enzyme, which is lipoic acid or lipoate. So whenever there is no lipoate, we cannot use pyruvate dehydrogenase enzyme. So what happened to that person? First of all, there is vomiting and diarrhea. There is skin abnormalities, white skin. There is QT prolongation because there is electrolyte imbalance, and also there is a garlic breath. So in first stage, very good mnemonic there, that the vampire, white skin vampire, is having diarrhea, watery diarrhea, and vomiting. And whenever it see a cutie girl have with the garlic breath. So this is all about the arsenic poisoning. So this is the next. There is also one point that alpha ketoglutarate also using pyruvate dehydrogenase cofactors, like all the cofactors which we have described in pyruvate dehydrogenase enzyme. It is also used by alpha ketoglutarate in the TCA cycle. So this MCQ is very much important. So this is all about how we found pyruvate and how pyruvate is converted into different four pathways.